Hello, can you hear me? Okay, so I think everyone is joined. So let's start now. Good evening, everyone. Let me brief you. Let's quickly revise what we did in last class. So we started with population ecology, and in population ecology, we studied that population is the number of species that inhabit a particular area during a particular time. So three things that we should be keeping in mind while reporting a population: that the number of individuals of same should be of same species in a particular area during a particular time. One thing that I missed in last class was that population is a genetic unit. Mm, Population is a genetic unit. That means, can someone tell me what is the unit for? 8 seconds. People are still joining. Okay. Yeah. So can someone tell me at which is the minimum level at which the transfer of gene is possible? Anyone? Can anyone tell me what is the minimum level of organization at which transfer of gene is possible? I think this question has come in your net exam. People are still joining. Mm, Yes, right. It's the species level. But species level is not called the genetic unit, it is the population level. Why so? Because in population we have all the collection of genes, that is all the genes or all the type of alleles that are present is basically collected, or the sum is in. Sir is saying something. Yes sir, yeah, it was in your oh, okay. Yeah, so population is called genetic unit because gene pool is the collection of all the genes which are present in the. I am entering her. I don't know why. There is some admitting problem actually. Hmm. Yeah. Oh. 1 second, guys. Please give me 1 second. There is some admitting problem. Few people are waiting, but they are not being admitted. I don't know why. One person is problem. Gunjan Kumari. What's I don't know. Okay. I have admitted, sir, but I don't know why it's not being admitted. It's flashing again and again on my screen. Okay, so yeah, the transfer of genes is possible in the species level. That means the gene can be transferred from one species to the another species also, and from one individual to the another individual of the same species also. And in case of population, we consider the collection of all the genes. That means whatever kind of alleles that may be present, that can be of the your heterogeneous allele, your yeah, your allele for dominant alleles, or your for recessive alleles. So these, or maybe if they are co-dominating alleles, or can be other alleles, like for the blood we have more than two alleles, and for some characters we have even four alleles. So हमें कैसे पता चलेगा कि एक पॉपुलेशन में कितने अलील्स हैं? तो जितने भी कलेक्शन होता है अलील्स का, वो एक पॉपुलेशन में ही होता है। किसी इंडिविजुअल में सिर्फ एक टाइप की अलील ही प्रेजेंट हो सकती है। तो इंडिविजुअल लेवल पे हमें नहीं पता चल सकता। इसलिए हम पॉपुलेशन को एक जेनेटिक यूनिट बोलते हैं, स्पीशीज को जेनेटिक यूनिट नहीं बोलते हैं। आई थिंक दिस इज क्लियर नाउ। Okay, if it's clear, let's move forward, because we are just revising for our last class. Okay, then we did that there are two types of organisms. First is the unitary, second is the modular kind of organisms. So if in this that I am revising, if anyone have any problem, you can ask now also. Okay, otherwise I'll start with the new things. And then we talked about the attributes of the populations that are applicable to only the populations, that is the whole group and not to the any particular individual. Then we talked about density, and we said that there are there are two ways in which we can find density. First is the numerical density, and second type in which we can calculate is the biomass density. Then again, there were two type of densities. First is crude density, and second is the niche density. I hope nobody has any problem in calculating crude and niche density. Yeah, so we then did that, the factors that affects the density and the application. Now, okay, so we have to start with the measurement of population density. So they are basically called as sampling methods in our terms. Okay, let's start with the first type of method, that is total count. In total count, we basically count all the number of individuals that are present in our area. uh, for example, this is mainly done for the plants. Like the number of trees in your area, you can manually count them also. You might have seen the markings on the tree trunks. If you have ever noticed in roadsides, along the roadsides, the trees are marked number one, number two. These are what was that? That was actually the manual counting of the number trees in particular area. Second, this can also be done for the animals which are larger in size, like your elephants, rhinos. They can be counted. टोटल काउंट कर सकते हैं उनका, फ्रॉम मे बी फ्रॉम एरियल सैम्पलिंग, एरियल व्यू से उनका टोटल काउंटिंग हो सकती है. द सेकेंड मेथड, second basic method for sampling is our line transect method. In line transect method, Yeah, in line transect method, what happens is the researcher has to travel along a line transect, which is basically randomly placed within the area. So there are two ways in which yeah, so there are two types basically, or I should say rather three types. First, when our area is not very dense, okay, there are very less vegetation in the area. The area is kind of desert or more or less semi-arid kind of area. In that kind of area, what we normally do is we just, like, this is our area which is bare, you can say bare, or maybe rocky semi-arid kind of area where having very less vegetation. We divide the whole area in such a way, means we plot our ट्रांजेक्ट्स, हम इस तरीके से ट्रांजेक्ट चुनते हैं अपने कि हमारा सारा एरिया कवर हो जाए. अब हम कितना से इस पूरे एरिया को कवर करने के लिए, से आई एम वर्किंग ऑन डियर. Okay, we are working on deer in a desert area. Now deer, हम deer का कितना हो सकता है home range? ज़्यादा से ज़्यादा, if it's a small rat deer, let's say इसका हो सकता है around 400 मीटर स्क्वायर, इतना ही होगा इसका home range. तो क्या हम क्या करेंगे, हम यहाँ पे दो ट्रांजेक्ट्स चूज कर लेंगे. ये दो ट्रांजेक्ट्स क्या करेंगे, इसमें 400 इधर 400 इधर, सेम सेम वे 400 इधर 400 इधर, हमारा पूरा एरिया कवर हो जाएगा. नाउ अगर हम ये दोनों ट्रांजेक्ट्स पे चलेंगे, हम अपना पूरा एरिया कवर कर सकते हैं. एंड वी विल, इसमें जो पॉपुलेशन डेंसिटी निकल के आएगी वो एग्जैक्टली वही आएगी जितने हमें नंबर ऑफ डियर्स दिखे हैं फील्ड में. ठीक है, इसमें हमें कुछ भी एक्स्ट्रापोलेट करने की, कुछ भी मैनिपुलेट करने की ज़रूरत नहीं है. अगर हमें इसमें 500 500 तो बहुत ज़्यादा है, 50 डियर्स दिखते हैं, तो हम 50 डियर्स रिपोर्ट कर देंगे कि वी हैव फाउंड 50 डियर्स इन आवर स्टडी. ओके, कुछ भी एक्स्ट्रापोलेट करने की ज़रूरत नहीं है. नेक्स्ट टाइप ऑफ एरिया इज दैट which is very dense, very dense area. If the area is very dense, we do not know exactly हमें कितना एरिया, हम मतलब कहाँ से ट्रांजेक्ट बनाए कि हमारा पूरा एरिया कवर हो जाए, क्योंकि विजिबिलिटी बहुत कम है, आर पार दिखता नहीं है. तो हम क्या करते हैं, हम उसमें एक ही ट्रांजेक्ट बनाते हैं और एक वो ट्रांजेक्ट एरिया के बीच में रैंडमली होता है. सो इवन इफ यू डोंट नो वेयर यू आर, मे बी यू हैव जीपीएस और समथिंग. सो लेट्स से यू आर वॉकिंग, यहाँ पे तुम लोग खड़े हो. अब तुम्हें यहाँ पे लगता है कि हम इसके अंदर जा सकते हैं, दिस इज यू सॉ सम पाथ काइंड ऑफ थिंग. तुमको फिर यहाँ से एक ट्रांजेक्ट सीधे बनाना है. ओके, इसमें एक चीज ध्यान रखा जाता है कि जहाँ से भी तुम जाओगे, यू हैव टू कट थ्रू इधर चल. So people basically जो vanishing cutters, the distance we take from both the sides कि हमें कहाँ तक देखना है, it depends on our animal that we are working on. अगर smaller animal है तो we take smaller area, जहाँ हम smaller area तक ही देखेंगे. अगर animal ज़्यादा है, ज़्यादा बड़ा है, तो हम दूर तक देखेंगे उसको. ठीक है, क्योंकि वो दूर दूर तक भी हमें दिख जाएगा easily. Okay, so yeah, basically you have to walk through it. Now, what is the technique in through which you calculate the population density from this method? Now look very carefully. This is the area. This is our line transect. We are we are moving in this direction. Okay. See, you are at this point right now. You saw an edge your compass. What you did here and see through the compass what is the, let's say it's 45 degrees, you find it is 45 degree. Sorry. Okay, you find 45 degree from the place where you are standing. I'll tell you how this degree is calculated. In your compass, okay, one second. Yeah, yeah. So in compass, let me choose a different color. This is your compass. This is north, east, west, and south. So the north direction always have 0 degree. ओके इन दी कंपास. कंपास में जो नॉर्थ डायरेक्शन होती है उसकी डिग्री हमेशा 0 डिग्री होती है. एंड यू विल हैव टू सी क्लॉक इन क्लॉकवाइज डायरेक्शन फ्रॉम नॉर्थ डायरेक्शन. ठीक है, तो तुम किसी भी डायरेक्शन में जा रहे हो, कंपास का जो नॉर्थ होगा वहां पे वो 0 डिग्री पॉइंट करेगा. उसके क्लॉक डायरेक्शन में तुम देखोगे कि तुम्हारा एनिमल कितनी डिग्री है. ठीक है, तो तुम ये by this method you will find the exact degree of your animal from where you are standing. उसके बाद उसके बाद this is the this di- distance from you. This is the distance of animal from where you are standing. Okay, this is your position. You this this is animal. Now we are more interested in knowing the perpendicular distance of organism from our line transect. So we are not this distance is called as radial distance, but we are not interested in this radial distance. We are interested in p, that is the perpendicular distance of the animal from our line transect, and this is the base b. Now you tell me which direction, which distance is voice breaking. Okay, let's wait for a few seconds. I think it was just a little fluctuation. Is it fine now, or is it still fluctuating? Fine now. Okay. Is my diagram clear to you, or shall I make a new? Okay, okay. I am repeat all over again. Are you guys able to understand? Do we see our? Okay, no problem. I'll repeat all over again. You first tell me. Is it? Are you guys able to understand that I told you about how to measure the angle, the angle of your animal with respect to you in the compass? No. Okay. Okay. So okay. So let me start all over again. Okay. Okay. So see, this is a compass. This is north, west, east, and south. What happens is in compass, the north correspond to always 0 degree. Okay, not always 0 degree. Followed by this east is your 90 degree, south is your 180 degree. This is two. Oh no. Okay, sorry, I cleared it by mistake. Okay. Yeah, so north always points at 0 degree. North will always 0 degree. So whenever you will stand at a point, you will see where is your north, and you will from north you will see clockwise. Okay, you will see clockwise wherever your animal is. Say you are here and you see your animal here. Okay, this is your animal. So from north your animal is at 90 till here, and let's say this is 20. So your animal is at 110 degrees. Okay, if your animal is here, here, so from north your animal is roughly at 45 degree, that you will see. So I was telling you how you are you going to look at the compass when you will follow a line transect. Okay, is it clear to you how do you look at a compass and know the exact degree at which your animal is present? Clear? Okay, is it clear to everyone? Please reply quickly, because I think I'm going very slow. Okay, so if it's clear how to measure the exact degree at which the animal is present, let's understand how to calculate the density using line transit. Okay, so I was telling you that this is your area. Say this is your study area, A. You made a line transect this, and now you are at the point here. You see your animal is here at this point. Okay, r is the radial distance. It's radial distance. This is you, this is your base, and this is the distance perpendicular. Yeah, so you took your compass and you measured this angle theta, that is the angle from you to that animal. Voice is breaking again. Okay, okay, let's wait for few a second. I don't know what's the problem. Hello. God. Frick. Is it fine now? I seriously have no idea why. Okay, give me 1 second. I just go and come. Uh, Hello. Am I audible now? I have just reconnected my broadband. I think there should not be any problem now. Yes. Okay. Yeah. Shall I start now? Yes. Okay. Yeah. So till now, are you able to get the concept till now? I basically talk that you are walking on the line transect, you are at this point and you saw the animal, and you from your compass you measure the angle at which it is present. Now you basically we are interested in knowing this p, that is the perpendicular distance from the animal to our line transect. Now we cannot measure this r, we cannot measure this radial distance, because from our line transect we are not going to go up to that point from here to here. No, it's not possible. What is possible is to measure this base. This base we can easily measure this base, because we are anyway we have to go further. So basically from here till here till this point we are going to measure this, how much is this. Then we'll going to we will apply our trigonometry. We know that this angle is 90 degree, as it is perpendicular drawn from this point till here. So ट्रिग्नोमेट्री में क्या होता है, p b p upon h h b. this is this is sine theta, this is cos theta, and this is tan theta. Now which is our hypotenuse. Our hypotenuse in this case is r, which is of course we cannot measure r. So basically we cannot use sine theta and cos theta here. We can, we are interested in knowing p and we can measure b, so we'll use tan theta. Okay, in tan theta, yeah, so in tan theta, theta is our this angle, tan theta is equals to perpendicular upon base. uh, are you all comfortable with the trigonometry and mathematical calculations? If anyone has any problem in mathematical calculation, you can tell me, I can repeat. Okay, no problem in that. Okay, so our tan theta is equals to perpendicular upon base, that is this upon this. Now say we traveled further and we found that b comes out to be 10 meter. Let's say b is 10 meter, and say our the angle theta, it was, we found that it was 45 degree. So what will be tan theta? It will be, I'm erasing it. Okay, yeah, so it will be 10 45 is equals to p upon 10, and 1045 is 1. Okay, the value of 1045 degree is 1, so p will turn out to be 10 meter. Okay, we'll find that p will be 10 meter by this method, and we are basically interested in p only. So is this clear to everyone how we calculated our perpendicular using this? Okay, and how can we calculate radial distance from our perpendicular and base? Now we know both perpendicular as well as base, so we will use what sorry the concept of Pythagoras theorem. Pythagoras theorem क्या होता है? h oh, sorry चलो कोई नहीं, h square is equals to p square plus b square, यही होता है ना? तो पाइथागोरस थ्योरम लगा के हम अपना आर भी निकाल सकते हैं, बट उसकी जरूरत नहीं पड़ती. ओके, हाँ, सो आई होप दिस इज क्लियर कि हमने फील्ड पे क्या किया. अब हम फील्ड पे गए, ये चीजें हम नोट करके ले आए. अब हम लेके आ गए, अब हमें करना क्या है? करना हमें ये है कि हमारा जो पॉपुलेशन डेंसिटी होता है उसका फॉर्मूला होता है n/2LA. So population density is equals to n, where n is the number of individuals we encountered. ठीक है, जो हमें इंडिविजुअल्स मिले पूरे लाइन ट्रांजेक्ट में के दौरान, वो सारे नंबर n होता है. दिस इज 2l. l इज द लेंथ ऑफ ट्रांजेक्ट, जितना ट्रांजेक्ट हमने ट्रेवल किया है उसकी लेंथ. एंड a, A is important actually. This is basically a half of effective strip width. इसका मतलब क्या होता है, ये एक कांस्टेंट है। ये कांस्टेंट जो है कैलकुलेट करना थोड़ा कॉम्प्लिकेटेड होता है, इस वजह से मैं तुमको पहली क्लास में भी बताना नहीं चाह रही थी, क्योंकि आई डोंट थिंक इतना डिटेल में हमें जाना है कि हम ए कैलकुलेट करेंगे। बेसिकली यू जस्ट अंडरस्टैंड दिस थिंग, इट्स अ कांस्टेंट एंड इट डिपेंड्स ऑन द साइटिंग डिस्टेंस। हमने वो पी जो परपेंडिकुलर था वो इसीलिए कैलकुलेट किया था ताकि उससे हम अपना ए कैलकुलेट कर सकें, क्योंकि ए जो है, ए इज, everyone has noted this formula, so can I clear it now? Yeah, uh, note this formula, note down this formula. ds, that is density, population density, is equals to n upon 2 la. Okay, so a is a, for a, just write it, it's a constant which tell us about the effective strip width, and it is directly proportional to the perpendicular distance of sighting. Okay, okay, let me, I myself write it. A is equals to constant which tell of the effective strip width, and it is directly proportional to the perpendicular distance of sighting. Okay, so we use basically various models which are developed by various scientists, and these models tell us, they, we just put the values that we you know the raw data values that we collect from the field, and we just put that in the models, and we get our a from, so by getting a we can then calculate our population density using the formula, previous formula that I told. Okay, so yeah, so various scientists have developed this the various models for calculating is a, like one that I remember is Hyan's model. Hyan's model. This model is developed for birds, flushing birds like grouse. उन्होंने ये मॉडल डेवलप किया था फ्लशिंग बर्ड्स के लिए। फ्लशिंग बर्ड्स, यू माइट बी नोइंग, सम बर्ड्स जो जब हम चलते हैं तो जैसे ही हम उनके थोड़ा सा पास जाते हैं वो चुप भाग के फटाफट झाड़ियों के अंदर घुस जाती हैं। उन बर्ड्स को कहते हैं फ्लशिंग बर्ड्स। ओके, सो आई थिंक I am done with line transit. If anyone have any doubt regarding line transits कैसे करते हैं और क्या क्या प्रोसीजर है, मेथड है, क्या मेथडोलॉजी है, you can ask me. तुम लोग को समझ में आ रहा है ना सबको? अब मुझे पता नहीं, बिकॉज तुम लोग जल्दी से रिप्लाई नहीं करते ना, तो मुझे डाउट रहता है कि पता नहीं है क्या पढ़ा रही हूँ, कैसे पढ़ा रही हूँ, समझ ही नहीं आ रहा है कुछ। Am I audible? Hello. What is the last thing that you that was audible to you people? I mean, I did not know what's going on. What's the last thing that you were able to hear? Yeah, about the model. So Heinemann model. Okay. So are you clear about the the all the things that I told you about the line transects and the methodology, the concepts, and how to calculate density?